Hello everyone, welcome back to my second Cellchat video tutorial. In today's video, I'm going to introduce the Cellchat databases, which are required for our cell-cell communication analysis using the Cellchat package. So the current Cellchat package has three databases. One is for the human ligand receptor interactions. If you are analyzing dataset from human, then you need to know the human database. The other one is for the mouse tissue. If you are analyzing mouse dataset, then you need to know the mouse database. So the zebrafish database was added in the Cellchat databases recently. If you're using zebrafish as a research model, and also you have zebrafish single-cell RNA sequencing dataset to analyze, so you need to know the zebrafish database. So we loaded three databases here. You can see the human, mouse, and the zebrafish. You can see each database has 4 elements. The name for 4 elements are the same in each database. So we can use the human database as an example to have a look at the 4 elements. You can see the first element is the ligand receptor interactions. The human database has 1,939 ligand receptor interactions. And the second element is the receptor or ligand complex. For the human ligand receptor database, there are 157 ligand receptor pairs have ligand complex or receptor complex. So the third element is the cofactor. In this database, there are 31 ligand receptor pairs has cofactors. Those cofactors are required to activate the signaling pathways. So the last element is the gene information. So in the human dataset is the human genome gene information. For the mouse dataset is the gene information for mouse genome, and for the zebrafish dataset, it is the gene information for zebrafish. So you can see in the human database there are 1,939 ligand receptor pairs. For the mouse dataset, you can see there are 2,019 ligand receptor base pairs, and for the zebrafish, it has 2,774 ligand receptor pairs. So we know the 4 elements for each dataset. Now we can have a look at the categories in each dataset. First, we can use the figure to show the human database category. You can see there are 3 pie charts. The first pie chart shows the percentage of different signaling pathways. So it has been divided into 3 signaling pathways. You can see in the human database, there are nearly 62% signaling pathways are secreted signalings, and around 22% interactions are between the ECM receptors, and about 65% are cell-cell contact. So in the human database, around 48% ligand receptors are heterodimers. And when they produced the human database, 73% ligand receptor interactions are obtained from the KEGG database, and 20% interactions were added from literatures. So let's clear this figure for the human database, and then we can promote the mouse database category because the figure looks very similar. So let's clear this one. Then we can promote the mouse database category. You can see it produces a very similar pie chart for the mouse category as the human category. It also has the percentage for the secreted signaling, ECM receptor, and cell-cell contact, and 47% are heterodimers. In the mouse dataset, 75% interactions were obtained from the KEGG database, and 25% were added from the literatures. So we can clear the plot for the mouse categories. Then we can plot the category for zebrafish. Again, you can see similar pie chart for the zebrafish database category. So I showed you the 4 elements for the human, mouse, and the zebrafish database. And also I showed you the categories for each database. So next we can use the human Cellchat database as an example to show you how we set the database for cell-cell communication analysis. So if you want to use the full human cell database to analyze your dataset, then you can set a Cellchat database.human as the Cellchat database use. If you run this code, you will set the full databases for human single-cell RNA sequencing data analysis. But if you are only interested in certain ligand-receptor interactions, for example, you are studying the secreted signaling pathways, then you can subset the database and only keep the secreted signaling pathways. If we run this code to subset the secreted signaling pathways, you will set the Cellchat database.use as secreted signaling pathways. If you have a look at the Cellchat data use at the moment, it is 15.5 megabyte. If we set it as secreted signaling pathways, then you can see it reduced to 15.5 megabyte. If you are studying the extracellular matrix protein receptor interaction, then you can subset the database as ECM receptor. If you are only interested in studying proteins that are required for cell-cell contact, then you can subset the database and only keep the ligand receptor interactions for cell-cell contact. If you run this code, you will set the database for cell-cell contact. The last part for the Cellchat database introduction, I want to show you how to update the Cellchat database. So I already show you each database has 4 elements: the interaction, complex, cofactor, and the gene information. So we can use the human Cellchat database as an example again to show you how to update the Cellchat database. So let's clear the data from here and also clear the plot from here. Then we can get the interactions from the human Cellchat database. You can see it has 1939 interactions. Then next we can get the complex, then cofactor, and the gene information. So now we can write CSV files for the 4 element. I'm going to write it into my folder Cellchat, then update as interaction CSV, complex CSV, cofact CSV, and gene information CSV. So let's open my folder for the update. You can see we have 4 CSV files: the interaction, gene information, complex, and the cofactor. So let's open the interaction CS file. This is the most important file that we need for our cell-cell communication analysis. So you can see there are 11 columns for the human Cellchat database. You see the first column is the interaction name. Here we can use the TGF beta as the example. So it's the TGF beta, then receptor 1 and receptor 2. The pathway will be TGF beta, and the ligand is TGF beta 1, TGF beta 2. The receptor will be TGF receptor 1 and receptor 2. So it also has a column for the agonist and another column for the antagonist. So some signaling pathways has the co-activator receptors. You can see here for the TGF beta signaling pathway, this column is empty, and the TGF beta signaling pathway has the inhibitor receptors. So the signaling pathway was obtained from the KGG database. For example, for this ligand receptor interaction, it was obtained from the publications. And another column called the annotations. That's what we see for the categories. You can see TGF beta is a secreted signaling pathways. Some pathways down there you will see probably the ECM receptors and also the cell-cell contact annotations. So the final column is the interaction name 2. So this is not very important. So now I hope you understand the information in the interaction files. So at the moment there are 1,939 ligand receptor pairs for the human dataset. If you want to study some ligand receptor interactions that were not included in the Cellchat database, then you can manually add your ligand receptor in this file to update the Cellchat database for your data analysis. So this is the CSV file for the interactions, and also we have the file for the cofactors. You can see in human Cellchat database there are 31 signaling pathways as cofactors. If you updated the interaction file with your ligand receptor interactions, and also your ligand receptor has cofactors, you can add your cofactors in this file for your signaling pathways. So the third file is the complex file. Again, it will be similar like the cofactor file. If the ligand or receptor in your signaling pathway has complex, then you can also update the complex file. So you only need to update the interaction, cofactor, and the complex CSV file. You don't need to update the gene information file because the gene information file has all the gene information. So for today's demonstration, we didn't add any new ligand receptor pairs into the interaction files, cofactor files, and the complex files to update the human Cellchat database. Suppose you added your ligand receptor pairs into the interaction file, then you can save the file with the updated ligand receptor interactions. Then we can read the updated files back into R again. First, we need to clear the cofactor, complex gene information, and the interaction files from here. Then we can read back the updated interaction file, the complex file, cofactor file, and the gene information. So now the files should contain your ligand receptor pairs, and we have 4 files loaded back into R again. Then we can make a Cellchat human database as updated list. You can see at the moment the list is empty. Then we can add the interaction back to the Cellchat database. Next, we can add the complex into the Cellchat database, then the cofactor, and finally the gene information. You can see now the updated Cellchat human database has 4 elements again. So you can use this updated Cellchat human database for your data analysis. What you need to do is just to set the updated human Cellchat database as Cellchat database use. Then you can use the updated cell database for your data analysis. Okay, I'm going to stop here for today's video. In my third video, I'm going to show you how to perform the interference of cell-cell communication network using the Cellchat package. So I hope to see you in my third video tutorial.